Okay, and welcome to the second part of our Chapter 11 lesson on meiosis and sexual reproduction. In this lesson, we're going to take care of these last four objectives, explain why meiosis 1 is called the reductive phase, describe the movement of chromosomes during both meiotic divisions, differentiate the events of anaphase 1 and anaphase 2, really important to understand what's going on there, and compare and contrast the major events of meiosis and mitosis. There are some similarities. So, want to just remind you of the basic structure of a chromosome. Now, remember that I call this a whole chromosome. To me, in my mind, a whole chromosome are two sister chromatids, which remember are genetically identical, connected at the centromere. All right, and they form this classic kind of X-shaped structure. That's a whole chromosome to me. These are sister chromatids when they are connected at the centromere. Now, during anaphase 2, when these come apart, when they separate, they are no longer chromatids, but we rename them chromosomes. Okay, but they're not whole chromosomes in my mind because it's not two chromatids joined together, okay? And so a quick overview of meiosis, and this is what we're talking about in this chapter, in this lesson of this chapter. So we have the diploid germ-line cell. Now remember, these are germ-line cells that we use to make ultimately, sperm and or egg, okay, or ova. And so it's a diploid cell, so this is a 2N cell. Now you'll notice that the chromosomes, we just have one arm of the chromosome here, but we have two sets, right? So in the case of humans, we have two sets of 23. And at this point, these chromosomes are genetically the same. Now, when we go through S-phase replication, we're going to double, or rather we're going to copy each of these chromosomes so that we end up with whole chromosomes. So we have the two sister chromatids connected at the centromere. All right, so you see what's going on there? We're doubling the blue. There's two blue connected together. And we're doubling the yellow. There's two yellow connected together. This is still a diploid cell. It's still 2N. All right. Now, in meiosis 1, then we're going to separate the homologs. And so we have this yellow guy going over there, this blue guy going over there. So now we go from diploid 2N to 2 haploid or 1N daughter cells. And that is why meiosis 1 is called the reductive phase, because we reduce the number of chromosomes in the parent cell to 1/2 in each daughter cell. So we go from 46 here in the case of humans, 46 to 23 here and 23 there. And then when you go through meiosis 2, that's where the sister chromatids are separated into the granddaughter cells. But now once these chromatids come apart and they're separated into their respective granddaughter cells, we don't call them chromatids anymore. These are chromosomes, just like up here, okay? It's kind of semantics, I guess, really, but that's the way it is. Okay, so we go from 2N to 2 1N daughter cells to 4 1N granddaughter cells. All right, and so this is just saying the same thing where the N number is 1, right? Through after interphase, after S phase replication, we have 2 chromosomes. So we have diploid chromosomes with sister chromatids, and after meiosis 1, see, prophase 1, metaphase 1, anaphase 1 to the phase 1. After meiosis 1, we end up with 1 haploid cell with sister chromatids because it has a whole chromosome in there. And then after meiosis 2, right, the chromatids separate. So we end up with a haploid cell. Okay, let's look, as we did with mitosis, the individual steps of meiosis 1. And so here's prophase 1. Now, think about what you know about prophase in mitosis. What happens in prophase of mitosis? Well, the nuclear envelope dissociates, the chromatin condenses so that we can physically see whole chromosomes, or chromosomes rather. Okay, that's what's going on here, and the asters form. The little spindle apparatus starts to form. Okay, so what's going on? In prophase 1 of meiosis 1, chromatin condenses, spindles form, homolog synapse. Now, and the nuclear envelope dissociates. The one thing that happens here in prophase 1 is crossing over, and we'll look at that in a little bit. But that's where homologs exchange genetic information. Actually, we talked about that in the first lesson. But this is where crossing over occurs. In metaphase 1, so what do you know about metaphase in mitosis? The chromosomes line up on the metaphase plate. They line up on the equator of the cell. Metaphase, that's what's going on here. The chromosomes, whole chromosomes, line up on the metaphase plate. We see it going on here, and we see it in the illustration here. Homologs is the line on the equatorial plate or the metaphase plate, and microtubules attached to the kinetochores. Remember, the microtubules here, the spindle apparatus, are like little ropes, right? And we connect them to the kinetochores, and we anchor them to the centrioles. And when we go into anaphase, we pull the chromosomes apart. They're like little ropes, and we pull the chromosomes apart. That's what that's all about, okay? In anaphase, what's going on in anaphase of mitosis? Chromosomes separate. You get one set going to one side of the cell, one set going to the other side of the cell. We have physical separation. Okay, and that's what you see going on here. Here's one set of chromosomes. Here's the other set of chromosomes. And these are whole chromosomes. So homologs are separated and pulled to opposite poles as microtubules shorten. So there's your centriole with the microtubules or spindle apparatus, and these are whole chromosomes. Now remember, this is after crossing over, so now they are mixed. Now we have genetic variability within the chromosomes themselves, but these are still whole chromosomes. And remember, a whole chromosome is made up of 2 sister chromatids. But now we have some genetic variability in there due to crossing over. So we get some elongation of the cell body going on here as well. And in telophase, the 2 cells begin to separate. We see that cleavage furrow, and nuclear envelopes begin to reform. So here we see that going on here. We have the set of chromosomes here, chromosomes here. You can see can't really see the cleavage furrow, but you can see the separation beginning there. But here you see it. The nuclear envelope begins to reassociate around these whole chromosomes now. Same over here. We see the cleavage furrow beginning to form there. So separated homologs cluster at each pole, nuclear envelopes form, and cytokinesis separates the two haploid daughter cells. Now, we've gone from diploid cells in prophase to now haploid cells. So we've gone from 46 chromosomes to 2 daughter cells, each with 23 chromosomes. And that's what we call meiosis 1 the reductive phase, because we reduce the number of chromosomes from 46 to 23. Okay, and that's it. Now we go into meiosis 2. Now those 2 daughter cells, haploid daughter cells that we made in meiosis 1, now those 2 cells go into meiosis 2. Think about what's going on. Now, think about just generally what's going on in mitosis, right? Chromosomes line up in the equatorial plate. The nuclear envelope disappears prophase. The chromosomes line up on the metaphase plate or equatorial plate. That's metaphase. The chromosomes separate, that's anaphase, and the nuclear envelopes reassociate, and we get the cleavage for the separation of cells. That's telophase and cytokinesis. So here's prophase 2. Now here we have our 2 daughter cells, haploid daughter cells with their mixed, potentially mixed whole chromosomes. Spindles reform, nuclear envelope begins to dissociate, chromatin condenses. So here we see chromosomes. Chromosomes here, you don't see the direct physical separation of these 2 cells, but the fact that you have 2 nuclear envelopes, you can see the line there, surrounding 2 sets of chromosomes, that tells you that these are 2 daughter cells. Now here's the deal. We like to describe the process here like going from telophase 1 to prophase 2. We like to think of it as in telophase, after anaphase and separation of the homologous chromosomes, the nuclear envelope reforms. You get this nice reformation of the nuclear envelope, and the cell divides, and you have 2 whole cells, and then the chromosomes unravel and you get chromatin again. Then you go into prophase 2, and the chromatin recondenses to form chromosomes in the nuclear envelope. Okay, it's nice to think of it that way, and that's fine and dandy if you want to really completely separate the 2 stages. But it's rare if at any time if you're making gametes, does a cell go from telophase 1 with its perfectly little formed nuclear envelope and chromatin is all spread all over the place, into prophase 2, where everything then has to be reversed, right? So if you're making gametes, then more than likely, or more often than not, the 2 daughter cells formed after telophase 1 will go straight into prophase 2 without the reformation of the nuclear envelope and without the unraveling of the chromosomes to make chromatin, because you don't want to spend the system, the body, the organism doesn't want to spend a lot of energy rebuilding nuclei and unraveling chromosomes and then turning right around and recondensing chromosomes and dissociating nuclear envelopes, right? It's much more energy efficient just to go from really essentially the end of telophase, the end of anaphase 1 directly into prophase 2. right? I mean, you still have to separate the cells, of course, but I'm talking about those details of the nuclear envelope and the chromatin, like that. It's really energy efficient not to go through those processes again. So everything the two cells separate, but all the chromosomes are still there, and there's no nuclear envelope there. All right, but when we describe it like this in textbooks, we like to separate it so that in your brain you can compartmentalize that and you know what's going on phase by phase by phase. But in real life, it's really just a kind of blurry transition, if you will. All right, so here we are in prophase 2. Nuclear envelopes dissociate. The whole chromosomes are there. We have our two daughter cells here. And then we go into metaphase 2. What happens in metaphase? Chromosomes line up on the equatorial plate or the metaphase plate or the equator of the cell. And here we see that. Chromosomes lining up. Now you have two cells going on here. There's the division. So just keep that in mind when you're looking at this micrograph. So we have an equatorial plate here, chromosomes lining up. You have an equatorial plate there, chromosomes lining up. Here we have our two daughter cells, the equatorial plate or the metaphase plate or the equator of the cell. The whole chromosomes are lining up. The spindle apparatus is connecting to those little kinetochores. Connected to the other end at the centrioles. Okay, chromosomes align on the equatorial plate or metaphase plate, microtubules attached to kinetochores of sister chromatids. Now remember, here we have whole chromosomes, but the idea is to get this down to 4 granddaughter cells. So here we have 2 daughter cells, and how can we make 4 granddaughter cells? Well, we have 2 chromosomes there, right? Or 2 chromatids in a whole chromosome. So we can separate the chromatids, and that's what happens in anaphase 2. Now here you see chromatids separating. Now once the centromere dissolves and the sister chromatids begin to separate, they're no longer called chromatids. We call them chromosomes. It's just one half of a whole chromosome, right? So we have these chromosomes going to this pole, these chromosomes going to that pole. We see the cleavage furrow beginning to form in both daughter cells. Here we see chromosomes there, so they're separating. Chromosomes here, clearly separated. That's anaphase 2. Anaphase 2 is, think of ana, against, antagonist, against. So you have sets of chromosomes going away from each other. And then in telophase 2, the cleavage furrow continues to form, the nuclear envelope reassociates, spindle apparatus disappears, and you end up eventually where these two cells separate, and that'll be 2 granddaughter cells, haploid, and these 2 cells separate, and that'll be 2 more granddaughter cells, haploid. So separated chromosomes cluster at each pole, the nuclear envelopes reform, cytokinesis separates the 4 haploid gametes. And so after meiosis 2, you have 1, 2, 3, 4 nuclei, even though we don't see clear separation of cells quite yet here, we do see 4 clearly differentiated nuclei. That tells you there are 4 new granddaughter cells there that we've just made. So the purpose of meiosis is to generate sex cells or gametes, sperm or ova or sperm or eggs. And so after cytokinesis of meiosis 2, we get 4 haploid gametes. So after cytokinesis, meiosis 1, we get 2 haploid daughter cells. After meiosis 2, cytokinesis, we get 4 haploid granddaughter cells, and we call those sperm on the male side, ova on the female side. Okay, so here we see it in humans, n equals 23 in humans, obviously, right? So the 2 in numbers 46 diploid, and as we go through meiosis 1, we keep it at 46 until we get to telophase and cytokinesis, and then we separate the 2 daughter cells, and now they are 23. They are haploid cells, but they're whole chromosomes, which means 2 sister chromatids making up each chromosome. We go through meiosis 2, and we end up with 4 we end up here with, okay, so we go from haploid from meiosis 1 into prophase meiosis 2. That's 23 chromosomes, 23 chromosomes here. Anaphase is 46 chromosomes because we have 2 sets of 23, so we have 46, but they're not whole chromosomes, right? They're just a single arm. And so then you end up, after telophase 2 cytokinesis, for, here it is, granddaughter cells, and they're haploid. It's only 46 here because at this point anaphase is just is 2 big cells, right? Each one with 23 chromosomes. Each one is haploid, not diploid. That's a little confusing right there. Okay, so this video is just a recap of the phases of meiosis. Meiosis 1 and meiosis 2. Pro, meta, anatelo. Okay, 1, pro, meta, anatelo of 2. Okay, it's just a recap. It's a nice recap. I think it's about 8 minutes long, worth your watching, just to kind of sum everything up. So meiosis yields variation, and going back to that first image that I showed you in part 1 of all the children, and then the children beside the image of the corn, and remember all of that variation comes via meiosis, and it's due to these things: random orientation of chromosomes at the first meiotic division. Okay, that's called the law of independent assortment. So there is nothing saying that chromosome, I'll just call them A, B, C, and D. Chromosome A has to go to the first cell, chromosome B over here, chromosome C over there, chromosome D over there. Okay, you could have chromosome A and chromosome D over here, and chromosome B and chromosome C, or you could have chromosome B and chromosome D over here, and A and C, like that. It's completely random how they get separated. Okay, it's called the law of independent assortment. We learned that from Mendel. And that's where variation comes in, because in one gamete you could have AB over here, and another gamete you could have AC, and another gamete you could have BA, and another gamete you could have CA, okay, like that. So it's completely random how they assort. It's called the law of independent assortment. It's completely random, and then so that yields variation, just the fact that they randomly assort. And then you have crossing over. Remember, crossing over in prophase 1 is where the 2 homologous chromosomes cross their chromatids and actually physically exchange genetic information. So you get genetic information exchanged between homologs. And then later on, in meiosis 2, you get another independently assorted group of chromosomes. So everything is mixed up when you get in meiosis 1, law of independent assortment, and then everything gets mixed up again with crossing over. Crossing over doesn't have to happen with every chromosome, but it does happen with many of them, and that's why you get over 8 million potential genomes for gametes. But so it can happen. And then in meiosis 2, then independent assortment happens again, and that's where more variation comes into play. All right, and this resulting variation, of course, is the fuel for evolution. Variation is the fuel for evolution, no matter, in all cases, in all organisms. Okay, so the law of independent assortment here. So maternal and paternal chromosomes from your parents are independently assorted. Okay, meaning that chromosomes from the same source do not have to end up in the same gamete. So you could have one gamete getting this one from mom, and this one from mom, and this one from dad, and this one from mom, and this one from dad, like that. And then you can have another getting this one from dad, and this one from mom, and this one from dad, and this one from dad, and this one from mom, like that. Completely independent, and it's completely random. That's the way evolution works. That's the way the natural world works. It's completely random. And so the number of possible arrangements for any given organism is 2 raised to the power of n, where n, of course, is the haploid number. So in the case of humans, right, n is going to be 23, and where n is the haploid number of chromosomes. Okay, so in this example, n equals 3, so the haploid number is 3. So 2 raised to the 3rd power. 2 cubed is 2 times 2 is 4, times 2 is 8. So there are 8 possible chromosome arrangements, 8 possible genome combinations for the gametes in this organism, right? And then here you see, and I'm not going to go through each one of these, but here's 8. And if you look at these closely, you'll see that each of these chromosome pairs is a little bit different from each one. So, but there's 8. Now, so the question is, how many chromosome arrangements are possible for humans? And I've already told you it's 2 to the n. N is 23 for humans, so it's 2 to the 23rd power. Get out your calculator, work that out, right? 2 times 2 times 2 times 2 times 2 times 2, 23 times. What number do you get there? It's a lot. It's over 8 million to give it away. And so over 8 million combinations possible every time a sperm is made or every time an ovum is made. Okay, so meiosis versus mitosis. So here we see in mitosis down here, okay, we have the parent cell, right, with its one-armed chromosomes. And so you're going to, in each case, you're going to go through a synthesis, interphase synthesis. And that's where you get whole chromosomes, what I call whole chromosomes. So in mitosis, here you have a parent cell with whole chromosomes. In metaphase, they line up on the equatorial plate or the metaphase plate. In anaphase, they separate. And in telophase, you end up with 2 genetically identical daughter cells, genetically identical to the original parent. Now in meiosis 1, same thing. You're going to go through interphase synthesis. You're going to make whole chromosomes here. This is your starting parent cell, 2n. Chromosomes line up on the equatorial plate. That's the same thing that's going on here. Whole chromosomes now. Chromosomes separate. Homologous chromosomes separate in anaphase, and then you end up with 2 1n daughter cells. Here you go from a 2N parent to 2N daughter cells, genetically identical. Here you go from a 2N parent to 2 1N daughter cells. You go from diploid to haploid. Now this is where mitosis stops, and then each of these cells can then go through mitosis. But of course, you know, in meiosis, these two daughter cells will proceed into meiosis 2. And so here we see those two daughter cells that we just made in the slide before coming over, and they're haploid, but they're whole chromosomes. So here we go into prophase 2. Chromosomes line up in the equatorial plate. Chromosomes line up on the equatorial plate for both of these guys, and then we go into anaphase 2, where now, right, these are whole chromosomes, so two sister chromatids apiece. Now the chromatids get separated. Once that centromere dissolves and the chromatids separate, now they are themselves chromosomes, and so we get from this daughter cell, which is haploid, 2 haploid gametes, and from this daughter cell we get 2 haploid gametes. I call these granddaughter cells because it's the next stage, but nevertheless, whatever you want to call them, gametes is better, really, but they're haploid. You get 4. Okay? Just a quick look, you can see the similarities between meiosis and mitosis. Meiosis 1, we go through DNA replication, 2N to 2N, diploid to diploid. Mitosis, we go through replication, right? This is the synthesis phase of interphase. And so we end up with a 2N, so we go diploid to diploid. Now in mitosis, this diploid cell will divide, making 2 diploid or 2N daughter cells, and that's where mitosis stops. And again, as many of you noted in your discussion response, mitosis is good for growth and development, repair of broken tissue. You get a cut, mitosis. You break a bone, mitosis, okay? Meiosis, the purpose of meiosis is to make gametes, right? This is the sexual reproduction contra to the asexual reproduction of binary fission. So after meiosis, so then we get the diploid to diploid, and then we go into meiosis 1, where this diploid cell gets divided. The whole chromosomes here, 46, get divided into 2 sets of 23. So we go from diploid to 2 daughter cells with a haploid number. And then these 2 haploid daughter cells will themselves go through meiosis 2, and you end up with 1, 2, 3, 4 haploid granddaughter cells or gametes. Just like in mitosis, errors can occur. We have this great identification and repair mechanism in mitosis, as you well know. In meiosis, we can get non-disjunction. Non-disjunction is a failure of chromosomes to move to opposite poles during either meiotic division. Right? They do not become they do not disjoin. That's non-disjunction. All right, so they do not come apart, and so you get a chromosome like you might get 21 chromosomes on one side and 24 chromosomes on the other, like that. And then there's aneuploidy, where gametes with missing or extra chromosome, or are gametes with missing or extra chromosome. So this is aneuploidy. This occurs at the end of meiosis 2, where you end up with gametes and they may have missing or extra chromosomes. And as the note here says, most common cause of spontaneous abortion in humans, because if you have, say, 20 chromosomes in a sperm and it fuses with an egg that has 23 chromosomes, and you only have 43 chromosomes, and a human needs 46 chromosomes to survive. If you only have 43 chromosomes or 44 chromosomes, then that zygote is not going to survive. It's not going to continue to replicate, because then it's going to go into mitosis, and the identification and repair mechanisms of mitosis are going to take it out. All right, that's how that works. Any questions? If you do have questions on this or anything else, you can message me 804-229-7993. You can email me jjeffries@rbc.edu, and you can also use the inbox here in Canvas. Thank you for your attention. See you next time.